We are Group 2, and we will be presenting our week 12 through 15 data to you today. First, I will be presenting data from our week 12 experiment. The purpose of this experiment is to isolate ZEB1 as a primary contributor to the regulation of ferroptosis sensitivity. Second, Jayan will present current research regarding our signaling protein Jun kinase, aka JNK, and the effects a JNK inhibitor is thought to have on cancer cells. Lastly, Maddie will be presenting the experimental procedure to test the effects of a JNK inhibitor with the broader goal of isolating the primary mechanism in which ZEB1 regulates ferroptosis sensitivity. Prior to the week 12 experiment from current research, which was confirmed by our own experiments, we know that the mesenchymal ZEB1 positive cells are correlated to ferroptosis sensitivity. However, the issue is that we do not know if there's a direct relationship between these two. We previously tested this on mesenchymal ZEB1 positive GS689Li cells derived from an individual clone of a single PC3 parental epithelial cell, so we can't confirm directly that ZEB1 was the primary and only factor playing a role in the ferroptosis sensitivity of these cells. Thus, our solution in the week 12 experiment was to more directly test ZEB1's role in ferroptosis by using an alternative cell line called TEM418 and by comparing our cells with ZEB1 knockout versions of those cells. So I'm going to briefly explain the highlights of the 3 cell conditions we will be using to manipulate our cells in this experiment. First is the TEM418 cells. These cells are an alternative mesenchymal subline of PC3 prostate cancer cells that have undergone an in vitro assay to select for cells with the ability to perform extravasation. In other words, these cells can migrate across an endothelial layer and thus have upregulated ZEB1 and downregulated E-cadherin expression. The ZEB1 knockout cells are cells that have undergone a ZEB1 gene deletion using the CRISPR method shown in the second image that you are all familiar with. This gene deletion results in a reversion of the mesenchymal ZEB1 positive cell phenotype to a more epithelial ZEB1 negative phenotype, in addition to the upregulation of the E-cadherin. I will be referring to the TEM418 knockout cells as epithelial knockout cells and the TEM418 cells as mesenchymal cells, which both reflect their phenotypes. Lastly, we will be using RSL3 to induce cells to undergo ferroptosis. As we know, RSL3 is a drug that inhibits GPX4, which is a protective factor for ferroptosis, as shown in this third image. Alright, so in this experiment, we used a 4-well plate containing cells with these 4 manipulated conditions. Cells are either TEM418 ZEB1 positive cells or the TEM418 ZEB1 knockout cells. And each of these cell types have been treated with RSL3 or untreated with DMSO. After sitting overnight, the cells are then imaged using 20x phase contrast and statistically analyzed using PRISM. In this experiment, we predicted that there is a direct relationship between ZEB1 and ferroptosis sensitivity. Thus, if our prediction is correct, we will expect that the ZEB1 positive mesenchymal cells treated with RSL3 to induce ferroptosis will have more cell death than the ZEB1 negative epithelial knockout cells treated with RSL3. In other words, with the induced ferroptosis from RSL3, ZEB1 positive equals cell death. ZEB1 negative equals cell survival, according to our prediction. So here are our representative phase contrast 20x images of our 4 treatment conditions. As expected, we see that the cell shape is a little more variable in the mesenchymal cells than the reverted epithelial knockout cells. We also see that the mesenchymal cells treated with RSL3 are very unhappy. They are small in size and abundance. We also see that both the treated and untreated epithelial knockout cells are happy and abundant, which is expected because both of them are the reverted epithelial cell type and neither of them should be sensitive to ferroptosis. So our last part of this experiment was to statistically analyze those images using PRISM and compare our results to our predictions. If our p-value is less than 0.05, then the variation in the data is significant and likely not due to random chance. So let's start out with the epithelial knockout cells. As mentioned earlier, both of these are reverted due to the gene deletion, thus they are epithelial in nature, so we expect And we see that there is no sensitivity to ferroptosis in either of these cell conditions, and no difference between the populations. Next is our big prediction, which directly compares our two cell types in treated conditions. We predicted that the mesenchymal cells would be much more sensitive to ferroptosis and cell death than their epithelial knockout counterpart. We see this is confirmed in our data with a very drastic difference between the two populations and a small p-value. For the mesenchymal treated versus untreated cells, we expected a difference since RSL3 is a ferroptosis inducer. Thus, the susceptible mesenchymal cells treated with RSL3 should have more cell death, which is confirmed in our data. Lastly, when comparing our two cell types untreated, we expect and confirm that the mesenchymal cell types have slightly more cell death than the epithelial knockout cells, since this is a general trend we see when we're looking at the strength of cancer cells in general. This experiment confirmed that there is a direct relationship and correlation between ZEB1 as a transcription factor and the regulation of ferroptosis sensitivity. Jayon will now talk about a specific pathway and signaling protein involved in ZEB1 activity called JNK.